The forest floor holds a medicine chest that no patent can touch. In 2012, a landmark study published in the journal Infection confirmed that allicin, the compound released when wild garlic is crushed, demonstrated measurable antibacterial activity against methicillin-resistant Staphylococcus aureus, the superbug that kills tens of thousands annually. Roman legions carried bundles of wild thyme across conquered territories, not for flavor, but because field surgeons documented faster wound recovery in soldiers who used it. By the late 19th century, thymol, extracted from wild thyme, was the primary active ingredient in Listerine, the world's first commercial antiseptic mouthwash. 5 plants grew wild across this land for millennia. 5 plants that healers, monks, and battlefield surgeons relied upon without question. Pharma replaced them with synthetic pills worth billions. And somewhere in that transaction, we lost something irreplaceable. This is not nostalgia. This is biochemistry. And before this video ends, one of these plants contains a compound so potent it was studied as a potential weapon against drug-resistant tuberculosis. Wild garlic is not the garlic of grocery shelves. Allium ursinum grows in dense woodland carpets across Europe and Central Asia, preferring the cool shadow of beech forests. Its allicin content is released only upon cell damage. Crushing, cutting, or chewing activates the chemistry. The compound is volatile by nature. It begins degrading within hours of exposure to air or heat. This instability is precisely why pharmaceutical synthesis of allicin remained commercially impractical for decades. You cannot patent a plant. You cannot stabilize wild allicin without destroying its core mechanism. A 2019 meta-analysis in the Journal of Antimicrobial Chemotherapy reviewed 19 clinical trials and confirmed garlic-derived compounds showed statistically significant activity against multiple bacterial strains. Wild populations contain measurably higher allicin concentrations than cultivated varieties. The industry had no interest in a cure that grew freely in every deciduous forest in the Northern Hemisphere. Nature had already solved the problem. The market needed a different answer. Thymol is a monoterpene phenol derived from the essential oil of wild thyme. Thymus serpillum colonizes rocky hillsides, dry meadows, and ancient stone walls from Scandinavia to the Himalayas. Medieval hospital records from the Abbey of St. Gall, dated to the 9th century, list wild thyme preparations among primary wound treatments. The science beneath that tradition is now measurable. Thymol disrupts bacterial cell membranes by integrating into the lipid bilayer, causing structural collapse. It operates against both gram-positive and gram-negative organisms. A 2011 study in the International Journal of Food Microbiology demonstrated thymol's effectiveness against Salmonella typhimurium and Staphylococcus aureus at concentrations occurring naturally in the plant's oil. Synthetic thymol was industrialized in the 1880s. Wild thyme became a footnote. The hillsides still grow it for free. Modern antibiotics cost what a week's wages once bought. The herb that preceded them costs nothing but the walk to find it. Carvacrol is the primary phenolic compound in wild oregano oil. Oreganum vulgare, the wild species, grows in rocky Mediterranean terrain, Anatolian plateaus, and mountain slopes across Central Asia. The cultivated oregano sold in supermarkets often contains less than 10% of the carvacrol levels found in wild harvested specimens. The distinction matters enormously. Carvacrol was the subject of over 800 peer-reviewed studies between 1990 and 2020. Research published in Molecules in 2013 demonstrated its mechanism. Carvacrol permeabilizes bacterial cell membranes, disrupting ion transport and causing cytoplasmic leakage. It showed activity against E. coli, Listeria, and Candida albicans in laboratory conditions. The pharmaceutical equivalent requires a prescription. The botanical original requires only dry, rocky soil and full sun. Wild oregano was listed in the pharmacopoeias of Greece, Rome, and Persia. It was removed from Western medical classification not when it stopped working, but when patentable alternatives became profitable. The mountain does not file patents. The corporation does. Wild horseradish is not a condiment. Armoracea rusticana. Escaped cultivation centuries ago and now grows feral along riverbanks, roadsides, and field margins across the entire northern hemisphere. Its biochemical mechanism involves glucosinolates, sulfur-containing compounds that convert to isothiocyanates upon cell damage. Isothiocyanates are chemically aggressive molecules. They inhibit bacterial enzyme systems by binding to sulfhydryl groups, effectively disabling the organism's metabolic machinery. A 2014 study published in Pharmaceutical Biology confirmed that horseradish root extract exhibited significant antibacterial activity against 12 clinical bacterial isolates, including antibiotic-resistant strains. Traditional European medicine documented horseradish preparations used externally for respiratory conditions throughout the 17th and 18th centuries. It grew along every river. It cost nothing. It required no laboratory. Pharmaceutical companies developed synthetic expectorants. Horseradish remained in the condiment aisle, its biochemical heritage reduced to a jar beside roast beef. The river still grows it wild. Purple coneflower was the most widely used medicinal plant among Plains Nations peoples of North America for over 4 centuries. Echinacea purpurea was documented by European botanists as early as 1699. By the 1930s, Echinacea preparations dominated American patent medicine markets. Then pharmaceutical antibiotics arrived. The American Medical Association declared botanical preparations unscientific. Echinacea was buried by institutional consensus, not by failed evidence. It was buried a second time when German pharmaceutical research in the 1980s began producing evidence of immunomodulatory activity, evidence that was inconvenient for synthetic drug markets. A 2015 Cochrane review analyzed 24 randomized controlled trials involving over 4,000 participants and concluded that certain Echinacea preparations may reduce the incidence and duration of the common cold. The immune system responds to alkylamides and polysaccharides in the plant's root and aerial parts. No synthetic compound replicates this action identically. And no patent can be filed on a wildflower that grows from Manitoba to Mississippi. The prairie still offers it freely. The pharmacy charges a premium for the echo. Natural compounds cannot be patented in their original form. This single legal fact explains more about modern pharmacology than any textbook. A pharmaceutical company invests an average of $2 billion to bring a synthetic drug to market. That investment demands exclusivity, a patent that prevents competitors from replicating the molecule for 20 years. Wild plants offer no such exclusivity. The return on investment from publishing research about wild oregano's carvacrol content is zero. The return from synthesizing a modified carvacrol analog, filing a patent, and marketing it as a novel compound is potentially enormous. This is not conspiracy. This is incentive structure. Medical journals in the late 20th century published fewer than 40 studies per decade on wild garlic's antimicrobial properties. The same journals published thousands on synthetic antibiotics during the same period. The science followed the funding. The funding followed the profit motive. The plants continued growing in silence, indifferent to the economics of their suppression. Wild garlic still carpets the beech forests of Europe every spring. Wild thyme still blankets the hillsides of Anatolia and the Alps in purple midsummer. Wild oregano still seeds itself into rocky Mediterranean soil without irrigation or intervention. Horseradish still pushes its enormous leaves up from riverbank mud in April. Impossible to eradicate, impossible to contain. Purple coneflower still stands tall on the open prairie. Seeds dispersed by goldfinches, roots deepening year after year. 5 plants. 5 biochemical systems refined over millions of years of evolutionary pressure. The antibiotic resistance crisis is not a natural phenomenon. It is the predictable consequence of replacing complex botanical chemistry with single-molecule synthetic drugs that bacteria learned to circumvent. The knowledge of these plants was not lost. It was set aside. Deliberately, economically, institutionally. Grow them. Study them. Remember what the forest offered before the pharmacy told us to forget.